name is Li Su, and I am a graduate student at University of Missouri. My talk today is Bioinformatics Analysis for Single-Cell Immune Profiling of Q fever vaccination in mice. So human Q fever is a worldwide zoonotic disease caused by the bacterium Coxiella bagnati with a high infectious rate. However, there is no FDA-approved vaccine. A previous study found that the formerly inactivated P1 vaccine, P1V, was more protective than the P2 vaccine, P2V, in guinea pigs. Our research objective is to explore the mechanisms of vaccine-induced protective immunity against C. bagnati infection. For this purpose, we set 3 vaccination groups. They are adjuvant and NM1 group vaccinated by P1V and the NM2 group vaccinated by P2V. And after the vaccination at day 7 and day 28, we collected cells and utilized 10x genomics immune profiling technology to simultaneously assay T-cell, B-cell receptor sequences, transcriptional profiles, and surface protein expression at the single-cell level. We first used WNN algorithm in CRAD package to integrate transcriptional profiles and surface protein expression, and then performed the cell clustering. Based on the auto-annotation given by single R, we manually clean and annotate all the cells into 8 major cell types. One of the hypotheses is that the P1V triggered a group of very unique immune cells that's responding to the pathogenic molecules, especially at the early vaccination stage. So we first moved to the B cells and performed the additional cell clusterings. We also used the Monaco 3 package to infer the pseudotime based on the assumption that adjuvant 7-day group was in the earliest development stage. Then we focused on those cell clusters that had a higher percentage of NM1 7-day vaccination group. Then we go back to see the pseudotime inference, and we found that those NM1 7-day higher clusters, for example, the cluster number 1 and the cluster number 18, were at a more advanced pseudotime. Then we also explored the VDJ gene usage in those NM1 7-day higher clusters, and we found that compared to the adjuvant groups, both the NM1 group and NM2 group had a more focused VDJ gene usage pattern, and between those 2 groups, there are still some difference in the VDJ gene usage preference. Then we moved to the T cells, and we also performed the additional cell clusterings. And after that, we focused on the CD4 plus T cells. And when we checked the VDJ gene usage in those CD4 plus T cells, we are quite exciting that compared to the adjuvant groups, the NM2 groups had a very similar gene usage preference, while the NM1 groups showed a very distinguished gene usage preference. So the take-home message for my research is that in part of the B cells and the CD4 plus T cells, NM1 group showed a very distinguished VDJ gene usage preference. This indicates that the P1V triggered a unique immune response and might explain why the P1V is more protective. This is all for my presentation today, and thanks for listening. If you have any questions or comments, you can leave me a message in the portal, or you can contact me via my email. My email address is lisu@mail.missouri.edu. Thanks very much.